Hello, welcome to chapter 24. We are going to talk about speciation. So by the end of this chapter, you should be able to compare and contrast the three common types of species concepts. You should be able to talk about the role that gene flow, selection, genetic drift, and mutation play in speciation. You should understand and be able to contrast the mechanisms of allopatric speciation and sympatric speciation, and then predict what happens when two divergent populations come back into contact. All right, so if you have two populations, like this one, and gene flow stops between the two populations, the allele frequencies are going to diverge over time. So the populations are going to evolve independently of one another, and this could happen for a bunch of reasons. It could happen because one population gets a different mutation that's not found in the other population. It could be because the environment is different and the natural selection is favoring different alleles in this population, and it could be due to genetic drift, meaning this side of the original population happened to have different alleles than this side, and as a result of these alleles being disproportionately represented, the allele frequencies change between the two populations. Okay, so this genetic divergence may eventually result in speciation. And speciation is a splitting event that creates two or more distinct species from an ancestral species. We know from empirical evidence that speciation can sometimes happen very quickly or happen very, very gradually over long periods of time. All right, so we define a species as an evolutionarily independent population or group of populations, okay? So great, that's a definition, you can memorize it, right? Okay, but how do you figure out if a lineage is evolutionarily independent, right? That's the hard part. So because, we have kind of an idea in our mind what we're aiming for when we're talking about a species, but we have to use, in practical terms, different kinds of criteria for deciding whether one group is two species or two groups are one species, etc. So I'm going to describe these three different kinds of criteria we use to figure out if you have a species or an evolutionarily independent population or group of populations. So we have the biological species concept, the morphological species concept, which is sometimes called the morphospecies concept, and the phylogenetic species concept. All right, so the biological species concept, the main idea is that you categorize species based on who they reproduce with. And so the idea is that if you have one group that is not able to reproduce with another group, you're not going to get gene flow between the two populations, right? And members of the populations do not interbreed, so that could be one way that the two populations are separate. Or it could be that they sometimes do interbreed, but when that happens, they don't end up making offspring that survive or can reproduce. All right, so what causes the gene flow between the populations to stop? Another name for causing the gene flow between populations to stop is called reproductive isolation. So it's like isolation between groups due to lack of reproduction. So we have kind of two categories for reproductive isolation. The first is called prezygotic isolation, and this means that you're not getting reproduction between groups because of something that's happening that's preventing the species from actually mating with each other, right? And then postzygotic, the species may be mating with each other, but the two groups are being kept apart because something happens after they mate so that the offspring don't survive or reproduce. So you can see from the name prezygotic, right? So that happens before a zygote is made. Hey you guys, what's a zygote?
It's the fertilized egg.
Yeah, it's a fertilized egg. So something happens before fertilization, and then postzygotic means after fertilization. So things can happen before fertilization that prevent the two different groups from merging into a single group, or postzygotic. All right, so here are the different kinds of ways that you have to cause reproductive isolation using the biological species concept. So you could have prezygotic isolating mechanisms like temporal mechanisms. So what's temporal mean?
Oh, it means it's related to time.
Yeah, related to time. So this means that the two populations are isolated because they breed at different times. So it could be different times of the day, different times of the year, you know. It could be even different years. So the example here is that bishop pines and Monterey pines release their pollen at different times of the year. It could be like you have spotted skunks and striped skunks. They breed at different times of year. You have different species of flies that some breed in the morning and some breed in the afternoon. And you have stuff that's even longer periods of time. Like, for example, we never have problems between different kinds of these 7-year cicadas breeding with each other because they don't reproduce but every 7 years. All right, you could have habitat that contributes to prezygotic isolation. So it could be that you have two populations that live in the same basic area, but not in exactly the same area. They're actually in slightly different habitats. So if you look along the edge of the beach, there's some areas that are grassy and some areas that are sandy, and you actually have one species that likes to live in the grassy area and one that lives in the sandy area. And because even though they're very close to each other, they never quite go into the opposite habitat. The sandy ones mate with the sandy ones, and the grassy ones mate with the grassy ones. All right, you can have behavioral mechanisms that contribute to prezygotic isolation. So often you have populations that live in exactly the same place, but they don't interbreed with each other because their courtship displays differ. So, for example, you have tons of male songbirds and female songbirds in one place, but each species, the male sings a very specific song, and in that species, the female only responds to the song that is sung by the male of the same species. You can have gametic barriers that prevent reproduction. So this could be because the egg and the sperm are incompatible in some way. This can be especially a problem in the ocean, right? So if you think of the ocean is just chock full of the sperm and eggs of a billion different sea creatures. And so you would wonder why you don't get different species sperm penetrating the wrong species egg. And this is usually dealt with chemically. So there is a bindin protein that is found in the sperm that when it meets up with the egg, if it isn't the correct form of the bindin protein, the egg will not let the sperm in. Okay, there's also mechanical reasons why you may have reproductive isolation prezygotically. It's a very cute little picture here of a Great Dane and a Chihuahua. So it's basically just saying that the matings fail because the male and the female reproductive structures are incompatible. They just don't fit together, right? They have large and small dogs, although I should point out large and small dogs are in the same species as each other. But you have other kinds of things, like, for example, insects. They have very specific genitalia, and so sometimes you have insects that fail to distinguish between members of one species and another species, and often their genitalia don't fit together correctly, so the male can't transfer sperm. All right, postzygotically, you can have hybrid viability issues. So that means that, the male and the female of the two different species get together and the male fertilizes the egg, but the offspring don't develop normally and usually die as embryos. So, for example, when ring-necked doves mate with rock doves, which can sometimes happen because they fail to identify each other as different species, less than 6% of the eggs from that hybrid crossing hatch. You can also have hybrid sterility. So this means that when you have breeding between two species, the offspring survive, but when they become adults, the offspring are not able to reproduce themselves. And I know horses and donkeys are domesticated animals, but this is an example that we can all kind of see around us. So horses and donkeys can mate and produce viable offspring, which are, I guess, either called mules, I'm trying to think how this goes. Female horse, male donkey, I think, is a mule. I think if you do it the other way, it's called a hinny. But mules, and I think also hinnies, are sterile and can't reproduce. Okay. So the biological species concept has some disadvantages. So, I mean, you can imagine, based on what I've just told you here, you could really go out in the world and find out if reproducing is happening, and then you would know, right? But sometimes you have species in which you can't look and see who's mating with who, like fossils, right? You don't know which fossils were mating with who when they were alive. Also, some species don't reproduce with anybody. So you have things like bacteria and yeast that reproduce asexually. So how can you tell what is a species of bacteria or what is a species of yeast? You can't use this kind of species concept to do that. You also can't apply it to populations that do not overlap geographically. So if you were looking at, for example, lions and tigers, right? They don't overlap geographically, so they would never have the opportunity to mate or not mate in the wild, right? Although we know that actually if you bring them into I don't know, whatever you would call it, a place where somebody would want to do that, they can actually reproduce. But it would never happen in nature because they would never see each other because of habitat isolation. All right, the second species concept is the morphospecies concept. And this is the idea that individual lineages are going to differ in their morphological features, meaning how they look, right? And that when you look at these differences, the reason why you have differences between the lineages is because at some point in the past, the two populations became separated from one another, and there was no longer gene flow between the two populations, right? So you're able to use these morphological clues to tell you that those populations have separate gene pools from each other. And this can be really useful when you don't have any data on gene flow, for example, because it's a fossil or something else. So you can use it on fossils and asexual species, and you can also potentially use it on sexual species when you need to. There are some disadvantages to categorizing species based on how things look. One of them is that you have some species that are polymorphic species. So what's polymorphic mean? What's poly mean?
It means many.
Yeah, it means many. What's morphic?
Morphic means form.
Yeah. So what would you guess polymorphic means?
It's like a species that looks different ways.
Yeah. So polymorphic species might be misclassified as more than one species, right? So some species have really diverse color patterns or maybe very different in size within the same species. And if you're not careful, you might accidentally categorize it as more than one species, even though the members of that species would consider themselves, they would mate with each other and there's no lack of gene flow. All right, so you also can't use the morphospecies concept to identify species that differ in non-morphological traits. Those are called cryptic species. So, for example, I used to work on salamanders, and there are some species of salamander that look exactly the same to us, but not to the salamanders. So now that we have access to DNA-based techniques, we're able to figure out that even though we're finding all of these salamanders in the same place, and they look the same, the salamanders don't think they look the same to each other, and they don't breed. There's two distinct species that only breed with themselves and don't breed with the other species. The third problem is which kinds of morphological features are important, right? So that can be subjective. So you would have to decide, okay, what's more important, whether you have double antennae or whether you have, an extra section in your body, okay? And if you choose wrong, you may incorrectly identify species. All right, the third concept is called the phylogenetic species concept. And so this identifies species based on evolutionary history. And the presumption is that all species are related by common ancestry, that's the assumption I think we can take. So on a phylogenetic tree, you're looking for a monophyletic group, okay? So I think this is the first time we introduced the idea of a monophyletic group. So mono means what?
It means one.
Yeah, monophyletic group. So phyletic is referring to kind of shared ancestry, usually, similarity, basically. So a monophyletic group consists of an ancestral population plus all of its descendants, okay? Another name for a monophyletic group is called a clade, or a lineage. We don't usually use the word lineage much, but you sometimes see this in like papers, peer-reviewed papers. Okay, so we will use the terms monophyletic group or clade. That means, again, an ancestral population plus all of its descendants. All right, so this is a phylogenetic tree, which we're going to talk about more in the next lecture. And what it's saying here is, okay, so this is the past, and this is the present over here. So you just imagine you're moving through time, And this is a population of individuals right here moving through time. And then at a certain point in time, this population of individuals has some event that causes them to split into two populations. So there's some lack of gene flow here. And then this lineage continues on, and then this lineage continues on over here, and then that new population gets split again into these other groups. Okay? So a monophyletic group is an ancestral population, and all of its descendants. So if this is the ancestral population, these are all of its descendants, right? If this is the ancestral population, these are all of its descendants. All right. This could be an ancestral population and these are its descendants, or this could be an ancestral population and these are its descendants, or this one over here, okay? All right, so far so good. I think we come back to this later, but I'll just say it because it's on this slide here. And you decide which groups are which based on synapomorphies, which is a trait unique to a monophyletic group. So this dash here is indicating this is when a particular trait occurred. Maybe it was fur in mammals, maybe it was loss of legs in snakes. It could be anything that is a trait unique to a monophyletic group. So that's just what that black line's showing. All right, there we go. So monophyletic groups are identified by synapomorphies. So, yep, a homologous trait. What does homologous mean? Anyone?
It was like similarities?
Yeah, similarities. Similarities why?
Oh, it was similarities because you inherited a trait from a common ancestor.
Yeah. All right, so homologous trait is a trait that two groups have in common because they inherited it from their common ancestor. Okay? So you want to figure out which groups are monophyletic based on homologous traits that are found in the common ancestor but missing in more distant ancestors, right? So you want to find something, a homologous trait that happened recently, not one that's a really, really old trait. So for example, if you want to categorize the mammals, you would say fur is a good synapomorphy in mammals, right? You probably wouldn't want to say something like having eyes. I mean, it's not really a synapomorphy in mammals because it's a trait that occurred a long time ago before mammals, okay? But fur is something that happened recently that mammals have and nothing else has. All right, so if we use this monophyletic species concept, a species is going to be defined as the smallest monophyletic group on a tree of life. So each of these lineages is a species. Cool? All right, let's try and think about synapomorphies for a minute. How about bird? Name a synapomorphy for a bird.
Feathers.
Yeah, feathers. Got any other ones?
Yeah, I got no teeth.
Yeah, you got no teeth. Loss of teeth. Yep. All right, does somebody have a synapomorphy for snakes?
Yeah, they only have one lung.
Yeah, that's a good one. They have lost one of their lungs. Anything else?
Yeah, they got no eyelids.
You're right, they have no eyelids. Anything else more obvious?
They lost their legs.
Yeah, they lost their legs. All right, name a synapomorphy for whales.
They got no fuzz.
Yeah, they got no fuzz. What else?
They live in the water.
Yeah, they live in the water. All right, anyone want to name a synapomorphy for some other group? I don't know, cat. Name a synapomorphy for cats.
Oh, I don't know.
Okay, that's fine. All right, let's move on. So, did you ever learn this in elementary school, that there's two main groups of elephants, the African elephant and the Asian elephant? You get the really big African elephants with the big ears and the Asian elephants with the little ears. Okay, I learned that when I was a kid. But actually, nowadays we have slightly different idea based on the phylogenetic species concept. So if you look at lineages of elephants, there's actually 3 different lineages. One that gives rise to the Asian elephants, and then 2 other lineages that give rise to the African forest elephants and the African savanna elephants. So we now consider these to be 3 different species based on the DNA sequences. Okay, so the phylogenetic species concept has 2 advantages. One, It can apply to any kind of population, fossil, asexual, or sexual. It's very logical because you would expect that two different species have different synapomorphies. And the synapomorphies arose because when the populations split from each other, they didn't have gene flow and they evolved independently, right? And part of that independent evolution is accumulating these synapomorphies. All right, disadvantages. Well, one disadvantage, which is becoming less and less relevant now, is that we didn't used to have phylogenies for all populations. Nowadays, it's becoming much more inexpensive to be able to get DNA sequence phylogenies for pretty much anything. So I would assume every once in a while you can't come up with a phylogeny for, all possible populations, but this is becoming less of a problem. Another disadvantage, which is more of a problem, is that using the phylogenetic species concept, you tend to recognize more species than any of the other species concepts. So for a long time, people were having debates between researchers they called lumpers and researchers they called splitters. Lumpers tended to lump different populations together and call them a species, and splitters tended to split different populations into their own species. So the phylogenetic species concept tends to result in more splitting. However, if that splitting is actually reflecting divergence between these different populations, then maybe we should actually, continue to do the splitting if it is actually biologically relevant. Okay, but people don't just pick a species concept. Usually researchers will use all three species concepts to be able to figure out evolutionarily independent populations in nature. But every once in a while you can come up with a conflict between a couple of different species methods that needs to be resolved. So here's an example of something. So dogs and wolves can potentially interbreed and produce fertile offspring. However, the wolf-dog hybrids are often not accepted into packs and can't find mates. So wolves and dogs are so genetically similar that DNA tests cannot distinguish one from the other. Would you say that wolves and dogs are one species or two species? So use species definitions to justify your answer. I don't know, Dog, do you want to start on this one?
I would not breed with a wolf.
Well, you are neutered, so that's a pretty safe bet.
Yes.
Okay, so, what do you think? Are dogs and wolves one species or two species?
They just look so different and behave so differently. I think they should be two.
Okay, so if you think they should be two because of how they look and behave, what would you call that?
That's the morphological species concept.
Yeah. All right, anyone else have other views? What about the fact that they can interbreed, but then the offspring don't do very well?
Yeah, I think that would be like biological species concept, that they would be two species, because that would be like some kind of post-zygotic reproductive isolating mechanism.
Oh, good one, Kat. Okay. So why do you think that?
Well, because the hybrid offspring will probably not make babies because they can't get into a pack.
Yeah. All right, and what about the DNA test that can't distinguish one from the other?
Well, so probably they're not distinct lineages on the phylogeny if DNA tests can't distinguish them from one another.
So what would that make them?
One species.
Yeah, so let's see, what do we have here? We have the morphospecies concept, says that it's two species, and the biological species concept says two species, but potentially the DNA tests say it's one species. So again, this is an example of a case in which we could spend a long time debating it. Okay. Let's look at an actual case in which the different species definitions were used. And so this is the case of the dusky seaside sparrow. Very handsome bird. I'm going to talk about subspecies here. So subspecies are defined as populations that live in a discrete geographic area, meaning they live in just this one place. They have distinguishing features, right? So they look a little different, but they're not considered different enough to be a separate species. And potentially, if you remove some of the geographic barriers that separate the different geographic areas from each other, then they can interbreed with each other. So it's kind of like if we say they're a subspecies, we're kind of saying, well, we think that they're part of the same species, but maybe speciation may be in action right now. We'll see what happens next. All right, so researchers have named 6 subspecies of seaside sparrow. So when you have a subspecies, so this is the genus name, Ammodramus, and then the species name is Maritimus, and then if you have a subspecies, that comes after this part. So like you have Ammodramus, Maritimus, Maritima, Ammodramus, Maritimus, McGillivrayi, I don't know, whatever. So this is, okay. But the one that is of interest is Ammodramus maritimus nigrensis, which is the dusky seaside sparrow. Okay? And so they live in these salt marshes along the Atlantic and Gulf coasts. All right, so these are the yellow ones right here. The populations are physically isolated from one another. These subpopulations are isolated from one another. And as you can see here, they differ in color, but they also sing different songs from one another. So in 1980, there were only 6 males left of the dusky seaside sparrow subspecies. Okay, and so biologists launched a conservation program under the Endangered Species Act. And the plan was, they figured that probably that population was most closely related to the one that was next to it. So they figured they'd breed those males with females from the nearby subspecies, and then they'd use the hybrids and reintroduce them into the area to reestablish a population that at least was preserving some of the genes in the dusky seaside sparrows. However, a phylogenetic analysis showed only 2 distinct monophyletic groups of seaside sparrows. So you have one group that includes the Maritima, the McGillivrayi, and the digrensis, and then the other one includes the Peninsulae, Juncalo, and Fisheri. Okay, so just the Atlantic coast and the Gulf coast ones were different from each other. So the dusky seaside sparrow here was genetically indistinguishable from the other 2 groups, but it was very different from these 3 groups. But unfortunately, when they did this breeding experiment, they bred those males from this group with the females from this group over here. They used the wrong population to try to preserve the existing genetic diversity, and consequently, the reintroduction program failed, and that subspecies ended up going extinct.
Why do all of the stories about birds end so long?
I'm sorry, I don't think they all do.
It kind of seems that way.
Okay. All right, so we're going to talk a little bit about how speciation actually happens. And to do that, we need to talk about biogeography. So biogeography, you know it as the bio part, living things, and the geography involving, I don't know, the earth. So it's a study of how species and populations are geographically distributed around the earth. And we have already talked about that you get genetic isolation between 2 populations when populations can become geographically separated, right? Something happens, some barrier prevents the populations from interacting. So when populations live in different areas, we say that they're in allopatry. So have you seen the word allo in the beginning of anything?
Like aloe vera lotion.
Yeah, that's a different allo. All right, if you haven't seen this, if you see allo in the front of a word, it means different, okay? And patry usually refers to kind of place of origin, right? So this just means different, place of origin. So populations that live in different areas, we say are in allopatry. And if you have speciation that starts with geographic isolation, we call that allopatric speciation. So there are 2 ways that you can get geographic isolation. The first is called dispersal, and the second is called vicariance. So in dispersal, so here's your original population, and then some of them move to a new habitat, so they colonize it and found a new population. Okay? When they get to the new place, they start to diverge genetically. It could be because they get a new mutation that these guys don't have, and that causes them to change. It could be that the environment is a little bit different and different alleles are favored here than were favored in the older place. It could be, what else? It could be genetic drift, so it could be by chance alone. The individuals that went over to this new place happen to have a greater proportion of maybe these pink alleles than in the original population. All right, eventually the populations diverge enough that they become genetically isolated from one another. So even if they could come back together again, they'd no longer be able to interbreed. Okay, so this is dispersal. Dispersal just refers to some of these individuals go somewhere else. Vicariance is when nobody goes anywhere, but something happens to the habitat that splits the groups from each other. So it could be, a giant crevice opens up in the ground and separates this population into two groups. After that happens, you have the same mechanisms that I just described with dispersal happening. So maybe this side has a higher proportion of the blue alleles, and so by genetic drift, you have more blues over here than over here. It could be that the environment is slightly different over here, a little drier or wetter, and the blue alleles are favored, so they increase over time. Or it could be that you get a new mutation that happens to be advantageous and it spreads over here. And then eventually the two groups become so different that even if they were to merge back together, they would no longer be able to interbreed. All right, so let's give an example of allopatric speciation by dispersal. So sometimes you get a colonization event that can lead to speciation. And we've already talked about Peter and Rosemary Grant, the cutest little elderly couple with their Darwin's finches on their islands. And what they did is they compared parents and offspring from large ground finch that remained on the island of Daphne Major. Okay, so those are the colonists, and those from the home island that came in, the migrants.
This story better not end up with more dead birds.
Well, we'll just have to see what happens. All right, so the colonists represented a new allopatric population, okay? But could that actually lead to speciation? So the average beak size in the colonist population was a lot larger from those of the migrant population, potentially due to genetic drift. Just the individuals that happen to, get there happen to have different beak size. Natural selection favored alleles associated with large beaks, and so the Grants concluded that the combination of genetic drift and natural selection contributed to the change in beak size between the two groups of birds. There's still a bit of gene flow, so the new population is not entirely separate from the other one, so we wouldn't really consider it a separate species, but it's something that we can keep our eye on, and maybe over time the populations will continue to diverge, and so we may be actually seeing speciation in action, but not complete speciation yet. All right, how was that story?
Okay, nobody died.
Okay. Here's another story where nobody dies about birds.
Great!
So vicariance can happen when you have animals that are just somewhere, and then geographic barriers appear that split the range of that animal into smaller ranges. So there's a kind of a bird called a trumpeter, and so 3 to 2.7 million years ago, this was their range, found over here in South America. And researchers compared the DNA sequences of trumpeters from the Amazon basin and lined it up with what they knew happened in terms of geological events. So around 2.0 to 2.7 million years ago, the Amazon River started to flow through here, and that divided the population of trumpeters into two populations, and those eventually became separate species, right? So this would be a vicariance, allopatric speciation by vicariance. Then the Amazon went this way and split again, and so that ended up producing 3 species, and then subsequent branchings of the Amazon between 0.8 and 0.3 million years ago caused 8 distinct species of trumpeters to speciate. All right. So you guys, tell me what vicariance is.
Well, it's when you have like a range where there's something living, and then a barrier comes down and like separates it, and then the species form after the barrier appears. Okay, so here's the Marion campus. So the biology building is new. It was kind of plunked down on top of this lot over here. All right, and there used to be a species of cricket that existed there, and then we put this building down on top of it.
And so if that event ended up splitting the crickets, what would that be?
That would be vicariance.
Yeah. So let's compare and contrast vicariance and dispersal. What are some similarities and what are some differences? Well, let's do differences first.
Yeah, so like in dispersal, the animals or plants or whatever are moving, but in vicariance they stay the same in the same place.
Yeah. All right, so what else? So that's the main difference. What's similar?
You need like natural selection and mutation and genetic drift to cause them to become different over time.
Yeah, that's true. Yep. Oh, there's a new building. Okay. So vicariance and dispersal are two kinds of allopatric speciation. So what does allo mean?
That means different.
Yeah. So have you seen sim on anything?
I think it means the same.
Yeah, Sympatric means same place, right? So populations or species that live in the same geographic area live in Sympatric. And researchers used to believe that speciation could not occur among Sympatric populations because they thought that if the two populations were living in the same place, gene flow would overwhelm any differences among populations that would be created by genetic drift and natural selection, so they didn't think that the populations could become different enough from one another to become separate species. But we do have studies that show that sympatric speciation can and does occur. And it occurs among populations in the same area. So we have here just an example. You have, a species that's living in one area, but for some reason you have divergence forming, okay? And eventually you end up with distinct populations. So maybe, over time you end up with just green reproducing with green and this gray color with gray color. And then the question is, what prevents gene flow between the two groups? All right, so we have two kinds of events that can cause the gene flow to stop. External events and internal events. So external events could be something like disruptive selection based on different ecological niches or mate preferences. So I'll give you some examples of that. And then internal events could be things like chromosomal mutations. All right, so you haven't seen the term niche before in this class, but a niche is the range of ecological resources that a species can use and the range of conditions that it can tolerate, okay? So you might have sympatric populations that are not geographically isolated, but they might be reproductively isolated because they might be adapting to different ecological niches by disruptive selection. So it may be advantageous to have this kind of trait value or this one, but not intermediate. So they may be some individuals specializing on this and some individuals specializing on this. And over time, they separate enough to become different species. So for example, in the United States, or North America generally speaking, there were hawthorn trees first, and then there were apple trees that were introduced, okay? And on the hawthorn trees, you have, hawthorns are like little tiny hard apples, right? And you have a fly called a hawthorn maggot fly, and it feeds and it mates on hawthorn. Apple maggot flies, we can see now around the US, feed and mate on apple trees. So the hawthorns were in North America first, then apples were introduced. So what we think happened is initially there were only hawthorn flies, but then when the apple trees arrived, some of the flies started to specialize on breeding on the apple trees. And so we can see that each of the species has a preference for its own host, okay? So apple flies, this is the percent of individuals that fly to a scent. So apple flies fly to the apple scent, hawthorn flies fly to the hawthorn scent. And so this makes it so the apple flies tend to be on the apple trees and the hawthorn flies tend to be on the hawthorn trees. We know that the ability of a fly to distinguish between the scents has a genetic basis, so it's basically genetically controlled. And we know that there are different alleles associated with the attraction to apple versus the attraction to hawthorn. Okay? When individuals are occasionally produced that are hybrids, or if you produce them in the lab on purpose, just for experimental reasons, they don't tend to orient to fruit scent as well as their parents do. So hybrids tend to have low reproductive success. They're less likely to find a fruit to make their babies on, and they're less likely to find mates. So hybrids tend to die off. So this is a situation in which you have to specialize, have a preference for either apple or hawthorn or a dye, right? So disruptive selection on choice of scent has led to sympatric speciation. Just to remind you, that's what disruptive selection is. All right. You can also have disruptive selection for mate choice, and this can cause rapid divergence among sympatric species. So, for example, fruit flies have strong mate choice. There's a fly that actually I do research on called Drosophila serrata, and they can live in both moist and dry habitats, and they express a particular gene called MFAS that causes them to synthesize a fatty acid that helps kind of coat their outsides and keep them from drying out. There is another species of nearby fly called Drosophila birchii, and this species does not express this MFAS gene. And because it doesn't express the MFAS gene, it can't kind of oil itself up to be able to live in a dry place. So birchii can only live in the moist rainforest part of Drosophila serrata's range. So MFAS does two things. It not only prevents their wings from drying out, it is also a chemical signal used in mate choice. So Drosophila serrata will not mate with Drosophila birchii because the chemical signal is not attractive to them, right? So the females do not like the smell of the males of the other species. So this is a prezygotic isolating mechanism. Do you remember which one?
Behavioral.
Yeah, behavioral prezygotic isolation. All right, I love that these fish are called porkfish. Anyway, these two species of porkfish first appeared as separate species 3 million years ago. Okay, the Isthmus of Panama arose 3.5 million years ago. Is this sympatric or allopatric speciation? Is it vicariance or dispersal? So what do you think?
Well, okay. So the Isthmus of Panama came up first, and then they became species, right?
Yep.
So it seems like it is allopatric speciation.
Yeah. And is the allopatric speciation by vicariance or dispersal?
Vicariance.
Yeah, vicariance. If you have sympatric speciation, do the terms vicariance or dispersal apply?
No, you can't have it. Just those are vicariance and dispersal are just about allopatric speciation.
Yeah.
I'd love to eat a porkfish.
Yeah, I know, Doug. All right, let's talk about another way. So we talked about one way that you can have sympatric speciation, right? What was that way?
That was like by disruptive selection. You can have disruptive selection because you like this tree, you don't like that tree, you like that fly, you don't like that fly, like that.
Yeah, sure. All right. The other kind of way you can have sympatric speciation is due to internal mechanisms. So you can have a mutation called polyploidy, which is very, very important to speciation. Does anyone remember, from 1113 what polyploidy is?
Yeah, that's like when you get extra chromosomes.
Yeah, like one extra chromosome?
I'm not sure.
So polyploidy is actually, you don't just get one extra chromosome, you get however many chromosomes you have, you get double that, okay? So you get like a whole new set of chromosomes. And this can happen when you have an error in meiosis or mitosis that gives you more than 2 sets of chromosomes. You're supposed to only have 2, right? One from your mom, one from your dad. Polyploidy means you have 3 or more sets of chromosomes. So you can get polyploidy happening in a couple different ways. One is autopolyploidy. So if you're an autopolyploid individual, so what's auto mean?
It means self.
Yeah, it means self. So this is when individuals have a mutation that doubles their chromosome number, but the chromosomes are all coming from the same species. So here's the parent, they're diploid, right? They have 3 chromosomes and 2 copies of each one. They have a non-disjunction event during meiosis. Do you remember non-disjunction from 1113?
Yeah, that's when the chromosomes stick together and end up like too many in one gamete and too few in another.
Yeah. Right, so you have a non-disjunction event here, and you end up somehow producing diploid gametes, okay? So instead of having one copy of each chromosome, you end up, I guess you probably have some gametes with no chromosomes in them, but those die, right? All right, so you end up with diploid gametes, and two of those diploid gametes then fuse with each other, and then you end up with tetraploid offspring. So those offspring have double the number of chromosomes of their parents, okay? And so you get an instantly new species, right? So if this happens to a plant, they can self, right? They can self-fertilize, they can breed with themselves, and so then they can go on and live as a new tetraploid species. Okay? They can't breed with the parent, or they wouldn't, I mean, parents are probably dead by now, but they can't breed with the parent species because you can't kind of, merge together gametes with different numbers of chromosomes. But they can reproduce with themselves. All right, so this is an example of instant speciation by autopolyploidy. Once researchers were out looking at maidenhair ferns, and they found a population of tetraploid maidenhair ferns just kind of in the middle of a normal population. So the parent obviously produced diploid gametes, and then those self-fertilized. So you ended up with like an instantly new species in the middle of another species. So those polyploid individuals were reproductively isolated from the parent population because they could breed with each other, but they were not able to produce sterile offspring breeding with the parent species. All right, so, yep, according to the biological species concept, you get speciation, and it happens very quick. You can also have a different way of polyploidy that results in sympatric speciation. You can have allopolyploidy. So, let's see. So what's allo mean?
I think it meant different.
Yeah, meant different. So you get an allopolyploid, and the different in this case means the chromosomes are coming from different places, okay? So this can happen when parents of different species mate, and there's some error in mitosis, and so you end up, instead of the offspring dying, which would be the most common case, you end up with viable non-sterile offspring. So for example, let's say we have two different parents. In this species, you have 3 chromosomes, 2 copies each. This one you have 2 chromosomes, 2 copies each. They undergo meiosis and make perfectly normal gametes, but then, just somehow, these 2 gametes fuse with each other, and you end up with hybrid offspring, and see, these red chromosomes and these blue chromosomes don't line up with each other, right? So you end up with 5 new chromosomes, right? So none of these pair up with each other. And so now you have a new species that is, that has 5 different kinds of chromosomes, 2 copies of each. Okay, so sometimes you can make new tetraploid species when 2 diploid species hybridize. Usually the offspring are sterile, but not always. So there's a couple of diploid species of this kind of flower, Trigopogon, introduced into North America, and they seem to have hybridized to produce this other species. Yeah. All right, so if you think that this kind of weird polyploidy event is like some weird freak thing that almost never happens, you're wrong. So many, many diploid plant species have closely related polyploid species. And so this suggests that there was some recent event in which the diploid species kind of split and formed a new species by polyploidy. Polyploids tend to have higher levels of heterozygosity than do diploids, which can be, let's talk about it. So how does heterozygosity affect average fitness in a population?
It's like better. It's better. If you're more heterozygous, you have better fitness.
Yeah. So polyploids do have higher fitness. They can self-fertilize more because they're not as affected by inbreeding depression as diploids. You also, if you have chromosomes that have been duplicated, so you get extra copies of chromosomes, then you kind of have some advantages. So some of those, if you have 2 sets of chromosomes, if you get a mutation in one set, you can just keep going. You still got the other set. And so this allows these duplicated chromosomes to diverge independently and increases genetic variation in the population. And let's see, so speciation by polyploidization does happen. You can get instant speciation, And this really high genetic diversity of polyploids has allowed rapid diversification of plants. So just showing pretty much every strawberry you eat now is a polyploid. When I used to do teaching labs in California, we would just use these for extracting DNA because they have so much DNA in them. These common flowers are all speciated by polyploidy, as are all of these crop items. All right, then finally, what happens if you have two isolated populations that come into contact with each other after they've been isolated for a while? Okay, so if divergence has happened and there is some kind of prezygotic isolation, then you're not going to get very much mating between the populations, and so you're not going to get much gene flow between the populations, and then the populations will stay separate from one another. But when prezygotic isolation does not exist, then sometimes you get the populations successfully interbreeding. And then you get gene flow between the two populations, and the populations may merge back together into a single megapopulation. But there are some other outcomes that I'm going to talk about. So, one outcome, right, they don't merge. Second outcome, they completely merge. And then some other outcomes, reinforcement, hybrid zones, and speciation by hybridization. So reinforcement is, if you have two populations that have diverged and are genetically distinct, you may end up with the hybrid offspring having lower fitness than the parents. So this could happen, for example, if you imagine that each of the two genetically distinct populations is adapted to a very particular area. If you produce a hybrid, that hybrid offspring may not be well suited for either habitat. When there's postzygotic isolation, there can be strong natural selection against interbreeding between distinct populations. And so there is natural selection for traits that prevent interbreeding among populations, and so that's called reinforcement. So if you have kind of behavioral mechanisms that allow species to distinguish one species from another and avoid mating with them, this would be a mechanism of reinforcement. And this is probably a trait that's arisen by natural selection because over time individuals who didn't have the ability to distinguish between their species and another species ended up having all offspring that died, and those and so the genes that help individuals distinguish between species spread over time. All right, so some of the best data on reinforcement comes from lab studies of Drosophila. And so we know that closely related Sympatric species very rarely mate with one another in the lab. So these are species that are caught from the same kind of area, right? Like they live in the same place, and they put them in the lab together and be like, all right, we're putting you together, mate. And they're like, no, not mating. If you take two species that are allopatric, meaning they live in different places, and you put them together in the lab, they will often mate, okay? And so this is a pattern you'd expect by reinforcement. The reason why individuals in this population won't mate with each other is because over time natural selection has selected for mechanisms that prevent the different species from mating with each other. In this case over here, where these two kinds of flies never actually meet each other in the wild, there's never been the opportunity for natural selection to work on any mechanisms that would cause them to avoid each other. All right, so sometimes hybrid offspring of diverged populations can mate and produce viable fertile offspring, and you end up with a hybrid zone. So that's a geographic area where interbreeding between the two populations occurs, and in that hybrid zone you tend to find hybrid offspring. So depending on the hybrid offspring fitness and how much breeding there is between the parental species, the hybrid zone could be very narrow or very wide, So I think maybe your book gave an example of like a hybrid zone between two species of crows that was like 30 miles wide because, crows fly really far. It could be long or short-lived. So sometimes, there's a short-lived hybrid zone that'll only be seen for a few years. In other places, there's hybrid zones that have been documented for hundreds of years between species. The hybrid zone could be exactly in one location, or it could shift over time. So I'll give you an example of that in a minute. So just a reminder for 1113, where did you get your mitochondrial DNA from?
From my mother.
Yeah, how come you didn't get it from your father?
Because it's in the cytoplasm, and the sperm doesn't have any cytoplasm. It's just like DNA with a tail on it.
Yeah, so all of us got our mitochondria from our mom, so we get our mitochondrial DNA from our mom, right?
Yeah.
Okay, so this is a case of a hybrid zone between Townsend's warblers and hermit warblers. So you can see here, these are the Townsend's warblers, and these are the hermit warblers, and this dotted, this line here is showing where their ranges overlap. And we can see when we look at them that the hybrid offspring look a little like each species. All right, using mitochondrial DNA, we can find out that most of the hybrids happen when Townsend warbler males mate with hermit warbler females. And we know this because, because the hybrid offspring have hermit warbler DNA, hermit warbler mitochondrial DNA. And so we think that what's happening is that the Townsend's warblers come into the hermit warbler territory, drive the hermit warbler male away, and then mate with the hermit warbler females that are on the nest. Okay. And so there's evidence that the hermit warblers, these guys over here, were once as far north as Alaska, but the Townsend warblers have taken over their range, pushing them farther and farther south. And so over time, this hybrid zone has moved farther and farther south. And then probably sooner or later, the Townsend's warblers will entirely drive the hermit warblers to extinction.
Okay, why do you keep telling us these sad stories about birds?
Well, I mean, it's only a sad story if you're a hermit warbler. If you're a Townsend's warbler, it's a great victory.
Yeah, I guess.
Okay, so if you have, what I mean by secondary contact is you had two populations, right? They become different from one another, and then later they come back together. You can get a range of different outcomes, so the two populations can fuse with one another, right? You could have reinforcement that causes them to stay apart. You could have a hybrid zone be founded. You could have one of the two populations go extinct. And you could potentially have them completely fuse together and form a new species. Or you could have them, they could fuse this way and be one species, or you could have a new species form from the hybrids in the middle. All right, so hopefully in this chapter, you've learned how a species is defined and identified, how species diverge, and what happens after the species diverge when the isolated populations come back in contact with one another.